So today our topic for the discussion is the tuberculosis. So as shown in the figure, the transmission of the TB or the tuberculosis is carried out by the infected respiratory droplets. You can observe here the infected individuals having the tubercle bacilli in the chest, and when he cough or they sneeze, it will transmit to the ear, and from the ear, the another susceptible individuals will carry in the lungs and able to produce the disease. So we can observe here the tubercle bacilli, the nature of the tubercle bacilli or the morphology of the tubercle bacilli. It is bacilli incept or the rod incept. So just I want to know, have you heard about the tuberculosis before? So you can pause the video, scan the QR code and answer it in the form of the yes or the no. So at the end of the session, you can able to discuss the general properties of the etiological agent, discuss the morphology of the Mycobacterium tuberculosis in brief, and describe the virulence factors that able to cause the disease, that is the pathogenesis of the Mycobacterium tuberculosis. So just you can pause the video and scan it to answer the questions that the which organ is primarily affected by the tuberculosis. So you can answer, you have options heart, lungs, kidney and the liver. So now we are going on a general introduction of the mycobacteria. So the mycobacteria are the group of the bacteria that belongs or that comprises the different species of the mycobacterium. So if we observe the general characteristics of the group, that is the mycobacteria, these, what kind of the bacteria it is? It is the rod shaped, or the nature, it is the aerobic, and it is the non-spore forming. And another property that is the important, that it resists decolorization by the acid or the alcohol. So it is also known as the acid-fast bacilli in the procedure of the Zn staining. So when we stain with the Zn stain, the bacilli resist the decolorization that is done by the or the carried out by the 20% sulfuric acid. So it cannot decolorize. So it is resist. So it is known as the acid-fast bacilli. And the most important pathogens to the humans among the group that the mycobacteria are Mycobacterium tuberculosis that causes the tuberculosis, Mycobacterium leprae that causes the disease that is the leprosy. Other bacteria also like the Mycobacterium avium complex, but they are less severe. So we'll discuss about the topic that is the Mycobacterium tuberculosis further. So here, etiological agent, Mycobacterium tuberculosis. So we are particularly confined to the single genus and the species that the Mycobacterium tuberculosis from broad. We have just read the Mycobacteria, that the group of the Mycobacterium, and we are confined to the Mycobacterium tuberculosis, the single agent or the causative agent that causes the disease known as the tuberculosis. So what kind of the bacteria it is? So it is the thin straight rods measuring about 0.4 to 3 micrometer in tissue when it is present in the tissue. But when we culture on the artificial media, it appears as a coccoid and a filamentous in form. Acid fastness, just we have discussed. It is acid fastness, so it is known as the acid fast bacilli. Why it is acid fastness? Due to the waxy envelope or the waxy cell wall that composed of the mycolic acid. So Zn stain, that is the Zell-Nelson technique, is the staining for the identification of the Mycobacterium tuberculosis. While when we consider the other bacteria, we stain with a gram stain, but the Mycobacterium tuberculosis is acid-fast bacilli because due to the presence of the mycolic acid in their cell wall. So we cannot stain with the general stain, that is the gram stain. We need the special stain, that is the Zn stain. So smears of sputum or the sections in tissue, if we observe under the fluorescence stain, if you stain with the fluorescent stain and observe, so we can observe as a yellow-orange fluorescence under the fluorescence microscope. And it is most preferred stains for the clinical specimens. So when we talk about the pathogenesis, just before we have talked about the morphology, the appearance of the Mycobacterium tuberculosis in the tissue form, in the artificial media, in the staining. How does it look like? So now the second one is the pathogenesis. What are the ability that it become the pathogens? So that is contributed by the different virulence factors. So in the previous or in the another bacteria, if we consider, so different bacteria have the different virulence factors. Some bacteria produce the exotoxins, some bacteria produce the endotoxins, some bacteria produce the different types of the enzymes. So in the case of the Mycobacterium tuberculosis, the lipids, the cell wall lipids and the different proteins, polysaccharides, so they are able to act as a virulence factors to make it pathogens. So let us discuss lipid that include the mycolic acid, waxes, and the phosphatides. The cell wall. The cell wall contains the lipids, and that lipids include the mycolic acid, waxes, and the phosphatides. Muramyl dipeptide, that is peptidoglycan, that is present in the cell wall, complex with the mycolic acid can cause the granuloma formation, which is one of the hallmark of the disease. Phospholipids induce the gaseous necrosis. So these muramyl dipeptide and the phospholipids that causes the disease in the lungs that acts as a virulence factors to produce as a disease or to appear as a disease in form in the lungs. Another is the proteins. Cell wall that contains the lipids. Second one is the protein that elicit the tuberculin reaction. So that for the diagnosis, we use the tuberculin test. Can also elicit the formation of variety of the antibodies. Polysaccharides contain a variety of the polysaccharides and can induce the immediate type of the hypersensitivity. And it also serves as the antigen in the reactions with the sera of the infected persons. So just we have discussed about the pathogenesis that is governed by the or carried out by the different virulence factors. The virulence factors are the cell wall that contains the lipids, proteins, and the polysaccharides. So how does it work? We can observe here. So we can observe here in the case of the pathogenesis, the bacilli during the coughing or the sneezing is released out and it is inhaled by the susceptible individuals by passing the physical barriers like the nasal hair or the we can say that respiratory secretions, that the mucous membrane, because the bacteria are filtered or bacteria are attached to the nasal hairs or they are trapped in the respiratory mucosa. And after the passing the physical barrier, they reach to the lungs. And in the lungs, they find the macrophage, alveolar macrophage. The alveolar macrophage will engulf that bacteria, and the bacteria, after the engulfment by the macrophages, the macrophage cannot able to kill it due to the inhibition of the phagosome-lysosome fusion. And the bacteria can survive inside the macrophage and able to thrive there, able to proliferate there, become a large in number, and then again release from the macrophage, and another macrophage will again engulf that bacteria, and the cycle is continued. So during the cycle of the engulfment of the macrophages and the multiplication of the Mycobacterium tuberculosis, the other immunological cells like the lymphocytes, neutrophils, natural killer cells, dendritic cells will came to the infected site. And after the coming to the infected site, they mesh together to form the granuloma. The process of granuloma formation will be started, initiated. So the macrophage that contains or trapped of the tubercle bacilli become the foamy one, and it is surrounded by the different immunological cells to form a special or the solid structure that is known as the granuloma. So after the maturation of the granuloma, the foamy macrophage, they will necrotize. And after the necrosis of the foamy macrophage, the bacilli will release from the foamy macrophage and remain as a inside the or the core of the caesium. So this is the caesium granuloma. So we can observe here the bacilli are present in the core of the caesium, and when the person becomes the immunocompromised by the different circumstances, either the HIV, malnutrition, diabetes, or other immunocompromised such circumstances, this caseous granuloma will rupture, and again the bacilli will release from the granuloma in the form of the coughing or the sneezing that contains in the respiratory droplets, and again the cycle is repeated. So this is how the tuberculosis will be produced in the human, and how it can be transferred from the one individual, infected individual, to the another susceptible individuals or the non-infected individuals. So we can observe here, immune cells are recruited to the wall of the infected macrophage, leading to the formation of the granuloma. That's the hallmark of the TB. Necrosis occurs, cheesy-like structures, that is the caseous formation. So upon the reactivation, the bacilli will proliferate, or the granuloma will rupture, and the bacilli will release from the granuloma and causes infection to the another individuals. So upon the granuloma rupture, disseminating the bacteria to the airways and causes infection to the another individuals. So you can observe here the granuloma that is formed on the lungs infected by the tubercle bacilli. So here, how the TB is transmitted. Again, you can pause the video, scan the QR code, or use the code and write the answer or click the answers. You have 4 options. So now we are summarizing the session, what we have read. That is, the Mycobacterium tuberculosis is the acid-fast bacilli, causes TB, and the most common infection to the lungs. Transmitted by the infected respiratory droplets, form granuloma, which is the hallmark of the TB, and primary infection that occurs at the base of the lungs. It infects the lungs, but in the case of the primary, it infects the base of the lungs, where in the reactivation cases, it infects the apex of the lungs. And for the reference of the suggested reading, you can go for this published article for your review. And please feel free to contact on the email address if you have any questions. And thank you for this session.